All right, we're now going to cover section 7 of chapter 2 on biological macromolecules. And after this unit, you should be able to identify the 6 chemical elements that generally compose biological macromolecules, describe a hydrocarbon and its chemical properties, and explain and give examples of functional groups and their chemical properties. Define the terms monomer, dimer, and polymer. Describe the role of water in both dehydration and hydrolysis reactions in altering biological macromolecules. Describe the general characteristics of lipid. Identify the 4 types of lipids and their physiological roles, as opposed to psychological, physiologic roles. Describe the distinguishing characteristics of carbohydrates. Explain the relationship between glucose and glycogen. Name some other carbohydrates found in living systems. And describe the general structure of a nucleic acid. Describe the structure of a nucleotide monomer. Distinguish between DNA and RNA. Name other important nucleotides. List the general functions of proteins. Describe the general structure of amino acids and proteins. So starting first with biological macromolecules, they are the large organ molecules that are synthesized by the body. They always contain carbon, hydrogen, and oxygen. Some may also have nitrogen, phosphorus, or sulfur. Carbon skeletons can take a variety of forms. Hydrocarbons contain only carbon and hydrogen atoms. They contain functional groups. Most are polar and able to hydrogen bond. Some act like acids, like carboxyl groups. Others act like bases, like an amine group, which includes nitrogen. And polymers are molecules made up of monomers, which are repeating subunits. Carbohydrates contain sugar monomers, nucleic acids contain nucleotide monomers, and proteins contain amino acid monomers. Dehydration synthesis or condensation occurs during the synthesis of biomolecules. One subunit loses a hydrogen ion, and another subunit, or hydrogen I should say, other subunit loses a hydroxide, and new covalent bonds are then formed and water produced. So you have the monomers here, and dehydration synthesis basically occurs, so you actually end up forming this covalent bond here between these 2 subunits. So what you end up with is basically an anabolic type reaction for dehydration synthesis. Hydrolysis occurs during the breakdown of biomolecules, where water is used, a hydrogen is added to one subunit, and a hydroxide added to another subunit. And so what you end up with then is breaking apart, basically into 2 units, breaking one molecule apart. And so that hydrolysis is essentially a catabolic reaction. So what functional groups may act as an acid? Carboxyl group could act as an acid. What defines a polymer? It's basically a stringing together of subunits, which are monomers. And the 3 biological macromolecules that are polymers and the monomers that compose them. Carbohydrates have their sugar monomers, and the nucleotides have their nucleic monomers. And of course, the proteins have their amino acid monomers. Lipids are a diverse group of fatty, water, insoluble molecules. They function as stored energy, cellular membrane components as phospholipids, and hormones like steroids. 4 primary classes: triglycerides, phospholipids, steroids, and eicosanoids. Triglycerides are used for long-term energy storage. They're formed from glycerol and 3 fatty acids. Those fatty acids vary in length and the number of double bonds. Saturated lacks double bonds. Basically, unsaturated is 1 double bond, so it's like basically polyunsaturated or 2 or more double bonds. Saturated tends to be rigid and flat in terms of actually their actual structure, whereas unsaturated is more as a kink structure, and they're basically in that kink structure, they are typically a liquid form unsaturated. Same with polyunsaturated. Okay. Adipose tissue stores triglycerides. Lipogenesis is the formation of triglycerides when conditions of excess nutrients exist. And lipolysis is basically the breakdown of triglycerides when nutrients are needed. And here we have an image of triglycerides, the hydroxyl groups, and the carboxylic acid. And lipogenesis basically occurs through the process of dehydration or dehydration synthesis, right? That's basically an anabolic type reaction. And then if you were going to have the other way around with triglyceride, lipolysis would occur through the process of hydrolysis, and it would actually break down a triglyceride, so you basically have a catabolic reaction. Phospholipids are amphipathic molecules that form chemical barriers of cell membranes. The phospholipid structure is similar to a triglyceride, except one end of the glycerol has a polar phosphate group. Glycerol, phosphate, and organic groups are polar. They form the hydrophilic head. or water-loving head. And then the fatty acid group is non-polar, form hydrophobic tails, and they form that phospholipid basically bilayer of the cell membrane, where you have the polar heads on the outer layers, and then you have basically the tails, the hydrophobic tails on the inner layer. And actually with hydrophobic basically intracellular fluid and the extracellular fluid outside the walls, because of that structure with the hydrophobic and the hydrophilic, with the polar heads on the outside and the basically non-polar tails on the inside, that's what's able to keep the intracellular fluid separate from extracellular fluid without something to help transportation across those cell barriers. Steroids are composed of hydrocarbons arranged in a multi-ring structure. 4 carbon rings, 3 have 6 carbon atoms, 1 has 5 carbon atoms. They differ in the side chains extending from their rings. Cholesterol is a component of animal plasma membranes, basically where steroids are formed from cholesterol. It's a precursor to other steroid synthesis. Steroid hormones like testosterone and estrogen are examples, where bile salts also, taurocholic and glycocholic acid for basically breakdown. And then, of course, here's an example of a steroid skeleton, looking quite similar to cholesterol because steroids are formed from cholesterol. Eicosanoids are modified 20-carbon fatty acids. They're synthesized from arachidonic acid, and they're a membrane component. They are local signaling molecules. Steroids in general are signaling molecules. The primary functions in inflammatory response, pretty common response for a steroid or sterol of any kind, sterol being a derivative from steroid, and nervous system communication. 4 classes of prostaglandins, eicosanoids. They control blood clotting and they cause fever and pain. You're actually trying to go against the prostaglandin response when you're taking an NSAID, okay, or anti-inflammatory. Prostacyclins reduce blood clotting. They cause blood vessels to dilate, and the opposite effect for thromboxanes, which are stimulating clotting and they're causing blood vessels to contract. They have different purposes for different times, right? And leukotrienes are basically, they sustain inflammatory reactions and allergic reactions. So lipid molecules typically dissolve in water. They do not. They're non-polar. Which class of lipids form cell membranes? That's the phospholipids. What characteristic allows it to perform this function? As we mentioned, essentially with the polar heads on the outside and the non-polar tails on the inside, makes it so you have hydrophobic structure on the inside, hydrophilic on the outside, so you can keep the extracellular fluid separate from the intracellular fluid, which is important, right? All right, we'll come to the 2.7c here in just a bit.